For most of recorded history, we told the story of exploration in a very particular way. Brave navigators set out from Europe, crossed uncharted waters, and arrived on shores that the rest of the world had never known existed. The names attached to those moments became legendary. Columbus in 1492, Cook in the Pacific, the Portuguese rounding Africa. These were the discovery stories, and for centuries they sat at the foundation of how we understood human civilization spreading across the globe. But science has a habit of complicating the stories we tell ourselves. And in the last 2 decades, a specific branch of science has been quietly dismantling the discovery narrative piece by piece. That science is ancient DNA analysis, and what it has revealed is extraordinary. Researchers can now extract genetic material from human remains, from animal bones, from plant samples buried for centuries, and read the molecular signatures left behind. Those signatures carry information about ancestry, about population mixing, about where a group of people came from, and who they encountered along the way. And when you apply that technology to remains found in places that were supposedly isolated until the age of European exploration, you start finding things that don't fit the old timeline at all. Genetic markers in South American populations that trace back to Polynesia, centuries before any European ship reached the continent's western coast. DNA in populations across the Pacific that tells a story of contact and mixing across thousands of miles of open ocean. Plant genetics that place certain crops on continents they were never supposed to have reached without human help. The evidence keeps accumulating, and it keeps pointing toward the same uncomfortable conclusion. People were moving across oceans far earlier, and far more deliberately than the standard history books ever acknowledged. Now, it's important to be precise about what DNA can and cannot tell us, because this is where the debate gets complicated. Genetic evidence can show that 2 populations share ancestry, or that a gene pool contains contributions from a geographically distant group. It can suggest when that mixing approximately occurred, using statistical models to estimate how many generations back the contact took place. What it cannot do is hand us a logbook. It cannot tell us exactly how many people made a journey, what their intentions were, or whether the contact was a single momentous voyage or a sustained series of interactions over generations. DNA gives us a biological signal. Reconstructing the human story behind that signal requires combining it with archaeology, linguistics, botany, and a great deal of careful reasoning. That distinction matters because ancient contact could have taken many different forms. There is a significant difference between a full migration, where a population relocates and establishes itself permanently in a new place, and a trading relationship, where goods and people move back and forth across established routes over long periods of time. And both of those are different again from a one-off voyage, a small group of sailors blown off course or deliberately pushing into unknown waters, making landfall somewhere unexpected and leaving only the faintest trace before the connection was lost. DNA can sometimes distinguish between these scenarios, but often it cannot. And that ambiguity is part of why these discoveries remain contested even among the scientists who uncovered them. There is also a deeper structural reason why maritime history is so difficult to reconstruct. The ocean leaves no road. A trade route through a mountain pass leaves behind waypoints, stopping places, worn stone, accumulated refuse. A sea route leaves almost nothing. Ships rot, ports get rebuilt. The people who sailed long distances in ancient times often belonged to cultures that kept no written records, or whose records have not survived, or whose records were never considered worth preserving by the literate civilizations that eventually documented them. The result is a systematic bias in the historical record. Overland contact is visible. Maritime contact is largely invisible, unless genetics or the movement of plants and animals leaves a trace that survives long enough for us to find. This invisibility has consequences. It means that for generations, scholars simply assumed that ocean crossings of any significant distance were impossible before certain technological thresholds were met. The Pacific was treated as a barrier rather than a highway. The Atlantic was considered uncrossable in practical terms, until the specific moment that European ships with European navigational tools arrived to cross it. These assumptions were not irrational, given the evidence that was available. But they were wrong, and the DNA record is forcing a reckoning with just how wrong they were. The case studies that have driven this reckoning are spread across the globe, and each one carries its own layer of complexity. Polynesian navigators who mastered the open Pacific centuries before European ships arrived anywhere near it. South American populations carrying genetic signatures that trace back across the world's largest ocean. The sweet potato, a crop native to the Americas, turning up in Pacific islands in pre-contact layers that no European trade route can explain. Chicken bones in Chile that may predate Columbus. Norse settlements in North America that archaeology confirmed decades ago but whose genetic legacy is still being traced. The Inuit and their extraordinary Arctic migrations. The peopling of Madagascar from Southeast Asia across thousands of miles of Indian Ocean. These are not fringe claims. They are published findings in peer-reviewed journals, and they are actively reshaping what we understand about human movement. But reshaping is not the same as resolving. Every one of these case studies remains contested in some respect. The dates are debated, the mechanisms are unclear, the degree of contact varies enormously from one case to the next. And some findings that seemed definitive when first announced have since been complicated by new data or alternative interpretations. What makes this moment genuinely significant is not that DNA has delivered a clean new version of history. It hasn't. What it has done is make the old version impossible to defend. The story of human exploration is not a story about Europeans arriving to connect the world. It is a story about ancient sailors, most of them nameless, navigating oceans that we once called uncrossable, reaching shores that we once called untouched, and leaving behind genetic echoes that only now, centuries later, we finally have the tools to hear. The question is what those echoes actually mean. And that is what we are going to figure out. To understand why any of the discoveries we're about to cover actually matter, you need a basic map of the tools scientists are using. Not because the genetics is complicated, but because the way these tools work determines what kinds of claims you can make and how confident you can be in making them. Get this part wrong and you end up either dismissing real evidence or getting excited about noise. So let's build the toolkit. Your DNA comes in several forms, and researchers use different types depending on what question they're asking. Mitochondrial DNA is inherited exclusively through the maternal line. Your mother passed it to you, her mother passed it to her, and so on back through time in an unbroken chain of female ancestors. Because it doesn't mix with paternal DNA the way most of your genome does, it stays relatively stable across generations, which makes it useful for tracing ancestry along a single lineage. Chromosome DNA works the same way but in the opposite direction, passing only from father to son, unchanged except for occasional mutations. Then there's autosomal DNA, which makes up the vast majority of your genome and comes from both parents, reshuffled and recombined with every generation. Autosomal DNA is the most powerful tool for detecting admixture, meaning evidence that two previously separate populations produce children together, because it captures contributions from all branches of a family tree, rather than just one line. Each type has trade-offs. Mitochondrial and Y-DNA are easier to recover from ancient samples and easier to trace through time, but they each represent only a single ancestral thread through an enormous family web. Autosomal DNA gives you a fuller picture but degrades faster and is harder to interpret cleanly. Modern genetic studies typically use all three in combination, cross-checking what each type reveals to build a more complete picture. Now, how do scientists estimate when populations mixed? The key concept is something called linkage disequilibrium, which sounds technical but rests on a fairly intuitive idea. When two populations interbreed for the first time, chunks of foreign DNA enter the gene pool in large blocks. Every generation after that, those blocks get broken up a little more through the normal process of genetic recombination. The smaller and more fragmented the blocks of foreign ancestry are in a modern population, the more generations have passed since the original mixing event. By measuring the size of those fragments and running statistical models, researchers can estimate within a range how many generations back the contact occurred. Convert generations to calendar years using average generation lengths and you get a rough date. Rough being the key word. These estimates carry wide confidence intervals, sometimes spanning centuries, and different models can produce different results from the same data. When you see a study claiming that populations mixed approximately 800 years ago, what they mean is that the most likely estimate clusters around that period, but the real answer could be significantly earlier or later. This brings us to one of the most important cautions in this entire field. Ancient DNA and modern population DNA are not the same thing, and conflating them causes serious errors. Modern population DNA is extracted from living people or recently deceased individuals. It reflects the current state of a gene pool, shaped by thousands of years of migration, admixture, drift and selection since whatever ancient contact you're trying to detect. Ancient DNA is extracted directly from old biological material, bones, teeth, hair, plant remains that were buried and preserved long enough to still contain readable genetic sequences. Ancient DNA lets you look at what a population's genome actually looked like at a specific point in the past, rather than trying to work backward from the present. That's enormously valuable, but ancient DNA comes with its own severe technical challenges. The most serious problem is degradation. DNA breaks down over time. In warm, humid environments, it can become unreadable within a few hundred years. In cold, dry conditions, it can survive for tens of thousands of years, which is why the best ancient DNA studies often come from Arctic sites or high-altitude locations. Even when ancient DNA does survive, it tends to exist in tiny, damaged fragments that have to be painstakingly reconstructed. And then there's contamination. Any modern human who touches a sample, excavates it, stores it, or processes it in a lab introduces the risk that their own DNA ends up in the results. The field has developed rigorous protocols to detect and eliminate contamination, including working in dedicated clean rooms, sequencing the sample multiple times, and comparing results against the DNA of everyone who handled it. But contamination remains a real and persistent risk, and some high-profile studies have been challenged or retracted because contamination wasn't adequately ruled out. Small sample sizes compound these difficulties. A single burial site might yield 1 or 2 individuals whose DNA can be recovered. Drawing broad conclusions about population-level contact from 1 or 2 genomes requires careful statistical reasoning, and the more dramatic the claim, the more caution is warranted. A genetic signal that appears in only one ancient individual could represent sustained contact between 2 civilizations, or it could represent a single person of unusual ancestry whose genome tells a personal story rather than a historical one. Sample size isn't just a methodological inconvenience, it's a genuine limit on what the data can support. This is why genetic signals almost always need support from independent lines of evidence. Archaeology, radiocarbon dating, isotopic analysis, linguistics, botany, and animal genetics can each provide confirmation or contradiction. A genetic signal suggesting contact between South America and Polynesia around 900 years ago becomes much more compelling if there are also plant remains from that same period showing the movement of crops between those regions, and more compelling still if archaeological sites on both sides show evidence of cultural exchange. When multiple independent lines of evidence converge on the same conclusion, confidence rises substantially. When genetics stands alone, the interpretation has to remain more tentative. There is also a logical trap worth identifying early, because it will matter for several of the case studies ahead. Absence of evidence is not evidence of absence. If researchers test a sample and find no genetic signal of contact with a distant population, that doesn't mean contact never occurred. The contact might have been too small, too brief, or too long ago to leave a detectable trace in the samples we happen to have. Preservation might have destroyed the relevant DNA. The sample might have been taken from a community that wasn't involved in the contact, while another community 100 miles away carries the signal clearly. The history of this field is full of examples where a genetic signal was absent in early studies and then found clearly once better samples, better techniques, or more sophisticated analysis became available. Absence is a reason for caution, not a reason for certainty. What this all amounts to is a picture of a genuinely powerful science operating under genuine constraints. DNA analysis can detect contacts that left no written record, survive in remote populations for centuries, and point researchers toward historical events that would otherwise be completely invisible. It has already transformed our understanding of human prehistory in ways that would have seemed impossible 30 years ago. But the confidence that headlines often attach to genetic findings frequently outruns what the underlying data actually supports. The best researchers in the field are careful about this. They publish ranges rather than precise dates, they note the limitations of their samples, they call for replication, and they frame their findings as evidence that requires convergence rather than proof that stands alone. Keep that framework in mind as we work through the specific cases. The evidence for pre-Columbian contact across the world's oceans is substantial, surprising, and in some cases genuinely astonishing. It is also, in almost every case, more complicated than the headline version suggests. The science is real, the conclusions are debated, and that combination is exactly what makes this story worth telling carefully. Before we get into the specific genetic signals and what they tell us about ancient contact, it's worth stepping back to ask a more fundamental question. Were ancient people actually capable of crossing vast stretches of open ocean? Because if the answer is no, then no amount of genetic evidence changes the picture. Extraordinary claims require extraordinary capacity, and the capacity has to come first. The answer, it turns out, is unambiguous. Long before European empires sent their fleets across the Atlantic and Pacific, human beings were navigating open ocean at distances that strain the imagination. This isn't speculation or romantic revisionism. It's one of the most thoroughly documented achievements in the archaeological record. And once you understand the scale of what was accomplished, the genetic signals we're about to examine stop looking like puzzles and start looking like natural consequences. The benchmark case is the Austronesian expansion. Roughly 5,000 years ago, a population of skilled seafarers began spreading out from the region of Taiwan and southeastern China. Over the following millennia, they colonized an enormous arc of the globe, from Madagascar in the west to Easter Island in the east, from Hawaii in the north to New Zealand in the south. At its fullest extent, the Austronesian world spans more than half the circumference of the planet. The people who built this world did not stumble onto nearby islands by accident. They sailed deliberately across stretches of open ocean with no land in sight, in some cases for weeks at a time, navigating with no magnetic compass, no GPS, and no printed charts. The vessels that made this possible were engineering achievements in their own right. Outrigger canoes and double-hulled canoes, sometimes called catamarans by later European observers, were designed specifically for the demands of ocean sailing. The double-hull configuration provided stability in heavy swells that would have capsized a European longboat. The hulls were carved and fitted with extraordinary precision, using wooden joinery that allowed some flex without compromising watertight integrity. Sails were rigged to catch wind efficiently and could be adjusted to sail closer to the wind than most people assume, giving navigators real options for managing direction rather than simply going wherever the wind pushed them. These were not rafts or dugouts carried helplessly by currents. They were capable vessels, crewed by experienced sailors who understood the sea in ways that took generations to accumulate. The navigation technology was equally sophisticated, though it left no physical artifacts. Polynesian and Micronesian navigators read the sky, the ocean surface and the atmosphere with a precision that impressed even the European sailors who encountered them centuries later. Star paths formed the backbone of the system. A navigator committed to memory the rising and setting points of dozens of stars across the horizon, knowing that each star, at its rise or set, points toward a specific direction. By tracking which stars rose where on a given night, an experienced navigator could maintain a heading through darkness without any instrument at all. During the day, the sun provided the same service, its arc through the sky shifting predictably with latitude and season. But stars and sun were only part of it. Ocean swells carry directional information encoded in patterns that persist even when wind and local waves obscure the surface. The major swell systems of the Pacific originate from consistent directions and can be felt through the hull of a canoe even at night, even in overcast conditions. Skilled navigators could detect the subtle bending and reflection of swells around islands they hadn't yet seen, sensing land through the motion of the water before it appeared on the horizon. Migrating birds, cloud formations that tend to pile up over land, changes in watercolor and temperature, even the smell of vegetation carried on the wind. All of these fed into a navigational system that was embodied rather than written, passed down through practice and memorization across generations. It worked. The settlement record proves it worked. Consider what that record shows. Remote islands like Hawaii sit more than 2,000 miles from the nearest large landmass. Easter Island sits in one of the most isolated spots on Earth, with the nearest inhabited island more than 1,000 miles away. New Zealand, which Polynesians reached around 700 or 800 years ago, required crossing the open Tasman Sea from the tropical Pacific to a temperate latitude. The Marquesas, the Tuamotus, the Society Islands, the Cook Islands, Hawaii, Easter Island, New Zealand, all of them settled by human beings sailing canoes before Europeans had any idea these places existed. Each successful colonization required not just a voyage but a productive colony, which means those canoes carried enough people, enough plants, enough animals, and enough genetic diversity to found a viable population. These were planned expeditions, not accidental drift. The timing of this expansion is well established through archaeology, radiocarbon dating, and now genetics. The settlement of the remote Pacific islands generally occurred in the first and second millennia of the Common Era, with some islands reached earlier and some later, but the broad pattern is clear and convergent across multiple lines of evidence. By the time Columbus was preparing his first voyage in the late 15th century, Polynesian navigators had already been crisscrossing the Pacific for 1,000 years. By the time European explorers began charting the Pacific in the 16th and 17th centuries, they were sailing into an ocean that had been a human highway for generations. Why does this matter for the question of pre-Columbian contact? Because it establishes beyond any reasonable doubt that the ocean was not a barrier. It was a road. People who could find Hawaii and Easter Island could find other things too. People capable of sailing from central Polynesia to the coast of New Zealand, tracking their position by stars and swells, were technically capable of sailing towards South America. The distance from the Marquesas to the coast of Peru is roughly the same as the distance from the Marquesas to Hawaii. Navigators who regularly attempted one of those crossings were not facing any fundamentally different challenge in attempting the other. This doesn't mean every proposed ancient contact actually happened. Capability is not the same as contact, and the fact that someone could have made a voyage doesn't prove they did. But it does mean the baseline assumption in this conversation has to shift. For a long time, the dominant framework in Western historical thinking treated the pre-Columbian oceans as impassable barriers that isolated civilizations from each other until European ships arrived to connect the world. That framework was never based on a careful assessment of ancient maritime technology. It was based on European ignorance of what non-European peoples were capable of. Once you accept that Austronesian navigators were doing things in the Pacific that European sailors wouldn't attempt for another 500 years, The question stops being whether ancient contact was possible and becomes a question about evidence for specific contacts in specific places. That's exactly where the genetics comes in. What researchers have been doing for the past 2 decades is looking for the fingerprints of those contacts in the DNA of living people and ancient remains. And what they found in some cases is striking. Not everywhere, not in every population, and not without complications. But in enough places, and with enough consistency across independent lines of evidence, to suggest that those Pacific highways carried more traffic than anyone in the 19th or 20th century was prepared to admit. The Austronesian expansion set the template. What came after it is where things get genuinely surprising. The previous section established that the Pacific was a road, not a wall. Now, let's trace exactly where that road went, and how far east it eventually reached, because the geography here is not incidental. It's the whole reason the DNA debate exists in the form that it does. The Austronesian expansion didn't move in one continuous wave. It advanced in stages, with populations pausing on islands for generations before pushing outward again. The Western Pacific was settled first, with communities established across Melanesia and the nearer islands of Micronesia thousands of years ago. But the deeper push into remote Oceania, the vast triangle of ocean that contains Hawaii, Easter Island and New Zealand at its corners, came later and came fast. Archaeologists and geneticists now have a reasonably clear picture of how that happened, and the timeline is more compressed than earlier researchers expected. The archaeological culture most closely associated with the initial push into the Pacific is called Lapita, named after a site in New Caledonia where distinctive pottery was first identified. Lapita people were sailing across open ocean stretches in the Western Pacific by around 3,000 years ago, establishing communities across Vanuatu, Fiji, Tonga and Samoa. These weren't the most isolated islands in the Pacific, but reaching them still required deliberate ocean crossings. Tonga and Samoa became a kind of staging ground, a place where Polynesian culture crystallized into the form that would later spread across the entire eastern Pacific. Populations there accumulated the navigational knowledge, the genetic diversity, the agricultural practices, and the canoe-building traditions that the later expansion would depend on. From Tonga and Samoa, the expansion eastward into the heart of remote Oceania began roughly 2,000 years ago, though the pace was uneven. The Society Islands, which include Tahiti, were settled probably by around the 1st century of the Common Era, possibly somewhat earlier. The Marquesas Islands were reached by a similar period, and these eastern Polynesian archipelagos became the springboards for everything that followed. What makes this phase remarkable is the sheer emptiness of the ocean being crossed. From Samoa to the Marquesas is roughly 3,000 kilometers of open water. There are almost no intermediate islands. A canoe heading east from Samoa would see no land for weeks before spotting anything that might support a colony. That crossing happened anyway, and happened successfully enough to found populations that persisted. The Marquesas appear to have been particularly important as a hub. From there, or from islands in the broader eastern Polynesian zone, voyagers reached Hawaii sometime around the 3rd to 5th century of the Common Era, though more recent archaeological dating has pushed the most reliable estimates toward the 9th or 10th century. The debate over exact dates continues, but the settlement was clearly accomplished well before the end of the first millennium. Hawaii sits at the northern apex of the Polynesian Triangle, more than 4,000 kilometers from the Marquesas, reached by navigators who had to cross not just distance but latitude, moving from tropical to subtropical waters through conditions quite different from anything in their previous experience. Easter Island, called Rapa Nui by its inhabitants, represents the eastern apex of the triangle. It was settled sometime around the 9th to 13th century of the Common Era, with more recent high-precision radiocarbon dating tending to push the estimates toward the later end of that range, perhaps around 1200. Easter Island's position on the map is worth pausing over. It sits approximately 3,500 kilometers off the coast of South America. That's a meaningful number. When Polynesian navigators arrived on Easter Island, they had placed themselves closer to the South American continent than they were to Tahiti. Whether they recognized that eastern landmass or made contact with it is precisely the question the genetic evidence will later address. But geographically they had already closed most of the distance. New Zealand anchors the southern apex of the triangle. It was settled last among the major Polynesian homelands, with dates converging on roughly 1250 to 1300 of the Common Era. The voyage required crossing the Tasman Sea and adapting to temperate conditions radically different from the tropical Pacific. Yet Polynesian navigators accomplished it within what is now a fairly tightly constrained window. The speed of that final phase of settlement, from the Marquesas and Society Islands to Hawaii, Easter Island and New Zealand, all within a few centuries, suggests navigational activity of considerable intensity during the period, roughly between 900 and 1300. The logistical constraints on Pacific voyaging were real and should not be minimized. These expeditions carried live plants for cultivation, animals including pigs, chickens and dogs, fresh water in sufficient quantities to survive crossings that could last weeks, and enough people to found viable colonies rather than simply perish on arrival. The canoes themselves required maintenance. Return voyages, which the evidence suggests did occur, required navigators to work against the prevailing winds and currents that had carried them east, meaning the western tack home was technically more demanding than the outward journey. Not every voyage succeeded. The Pacific almost certainly swallowed canoes and crews we will never be able to count. The settlements we can document represent the successful fraction of a much larger pattern of activity, and yet the opportunities were equally real. The Eastern Pacific has a structure that rewarded experienced navigators. The trade winds blow generally from east to west, which favoured outward exploration on a return-capable basis. A canoe could run east with modified wind strategies and return west more easily on the trades if needed. The intertropical convergence zone creates seasonal variability that skilled navigators could exploit for different headings at different times of year. Ocean currents, swell patterns, and bird migration routes all provided information that could guide a vessel without any instrument more sophisticated than accumulated human attention. The same capabilities that allowed Polynesians to settle Easter Island in the first place also put the coast of South America within theoretical reach of a crew willing to push east from that final island. This is why remote Oceania and eastern Polynesia in particular sits at the centre of the ancient contact debate rather than at its edges. The region is not just a backdrop. It is the geographic reason why contact between Polynesian voyagers and South American populations is a plausible hypothesis rather than a fantasy. The chronology matters too. By the time Easter Island was settled, by the time Hawaiian culture was developing its mature forms, by the time New Zealand was being colonized, Polynesian navigators had been refining their techniques for 1,000 years. They were not novices bumbling toward an unexpected continent. They were the most accomplished ocean voyagers in the world at that moment in history, operating at the peak of a tradition built over dozens of generations. When geneticists began looking at the DNA of Polynesian and South American populations and finding signals that seemed to cross the ocean in both directions, the Eastern Pacific timeline gave those signals a plausible mechanism. The voyages weren't theoretical possibilities requiring imagination. They were a demonstrated pattern of behaviour applied to a region where the distances involved were no greater than crossings that had already been made multiple times. The question was never really whether the canoes could reach South America. The question was whether anyone was on them when they did, and that is exactly what the DNA was being asked to answer. So the canoes could have made the crossing. The geography allowed it, the navigational tradition supported it, and the timeline placed some of the world's most capable sailors within a few weeks of the South American coastline. But capability is not the same as contact. For decades, the question of whether Polynesian voyagers actually reached South America, or South Americans actually reached Polynesia, remained in the territory of intriguing hypothesis rather than verifiable fact. Then the genetics arrived, and the conversation changed register entirely. The most significant findings came from a series of studies analyzing DNA from present-day Polynesian populations, particularly those living on islands in eastern Polynesia. What researchers found was a distinct genetic signature that did not fit neatly within the Polynesian ancestral profile. Alongside the Austronesian heritage that connects Polynesian populations to their roots in island Southeast Asia, there was another component, traceable not to Asia or Melanesia but to the indigenous populations of South America. The signal was not enormous in proportional terms, but it was consistent, it was statistically robust, and it pointed in a direction that the navigational history had already suggested was worth looking. The populations in which this signal appears most clearly are concentrated in eastern Polynesia. The inhabitants of the Marquesas Islands carry it. Populations on several of the Tuamotu atolls carry it. Communities in what is now French Polynesia more broadly show versions of it. Perhaps most discussed among researchers, the people of Rapa Nui, Easter Island, carry a Native American genetic component that has now been identified in multiple independent studies using different methodologies. The geographic clustering matters here. These are exactly the communities you would expect to show a South American signal if contact happened in the Eastern Pacific, which is the only place where the distance between Polynesia and South America becomes bridgeable by pre-industrial watercraft. The signal is not distributed randomly across the Pacific. It concentrates in the places closest to the American continent, and that pattern is itself a piece of evidence. Estimating when the admixture occurred requires statistical modeling of how genetic variation accumulates and breaks down over generations, a method known as admixture dating. Different studies have produced slightly different estimates, but the results have converged on a period somewhere around 1200 CE, give or take a century or two in either direction. That timing is consequential for two reasons. First, it aligns closely with the archaeological evidence for when eastern Polynesia, including Easter Island, was being actively settled and developed. Second, and equally important, it places the mixing event well before European contact with the Pacific, which in this region began seriously in the 16th and 17th centuries. The dates do not describe a medieval curiosity that might be dismissed as later colonial disruption. They describe a pre-contact phenomenon. That distinction matters enormously because the most obvious alternative explanation for Native American DNA in Polynesian populations is that European colonialism reshuffled populations across the Pacific in ways that were violent, coercive, and extraordinarily disruptive. Indigenous people from the Americas were transported across oceans by colonial powers. Forced migration, enslavement, and the demographic chaos of the colonial period all created opportunities for genetic mixing that had nothing to do with ancient voyaging. If the Native American signal in Polynesian populations dated to the 18th or 19th century, it could be attributed to these processes without much controversy. The admixture dating research argues that the signal is older than that, old enough to predate the Europeans entirely, and that is what elevates it from interesting artifact to potential proof of ancient contact. Researchers also work to rule out colonial-era mixing through the structure of the signal itself. When admixture is recent, the chromosomal segments inherited from the outside population tend to be long, because recombination has not had time to break them up. The older a mixing event is, the shorter those segments become as they are shuffled across successive generations. The Native American segments found in eastern Polynesian genomes have characteristics consistent with having been present for many generations, not the relatively intact long segments you would expect from mixing events only a few centuries old. This is not a foolproof method, and researchers acknowledge the statistical uncertainty involved, but it adds a layer of analysis that goes beyond simply detecting the presence of a signal. It addresses the question of when that signal entered the population. What the genetic evidence does not tell us is the nature of the encounter itself, and this is where precision becomes essential. Finding Native American DNA in Polynesian populations demonstrates that biological reproduction happened between people from these two groups at some point before European contact. It does not specify whether Polynesian voyagers reached South America and returned, whether South Americans crossed the ocean to Polynesia, or whether contact occurred somewhere in between, perhaps on an island that no longer supports population or that has been overlooked. It does not tell us how many people were involved, whether the contact was peaceful or hostile, repeated or singular, intentional, or the result of drift. It does not describe a cultural exchange, a trade relationship, or a sustained connection. It describes a moment, or a series of moments, in which the gene pools mixed. Everything beyond that is inference built on additional evidence from archaeology, linguistics, and botany. This is one reason some scholars remain cautious about how the results are framed, even when they accept the signal itself as genuine. The jump from there was genetic admixture to there was intentional contact to Polynesians discovered South America involves steps that the DNA alone cannot fully support. A small number of individuals arriving by accident, surviving briefly, and contributing to a local gene pool would produce a signal in the genome that looks identical to a sustained voyage of exploration. The genetic record does not preserve motive or method. Researchers who accept the admixture finding as solid therefore often differ considerably from each other on what story it actually tells. There is also ongoing discussion about which South American population contributed the genetic material. The Amazonian and coastal populations of South America are genetically distinct from each other and from the various indigenous groups across the continent's geography. Early studies pointed toward populations with genetic affinities to present-day Colombia or nearby regions, though the resolution of ancient ancestry analysis has limits when the reference populations available are modern ones that have themselves experienced centuries of mixing and displacement. More refined analyses have continued to debate the precise origin point on the South American side, which affects the plausibility of different contact scenarios, since not all South American populations were equally positioned geographically or culturally to make or receive an oceanic crossing. The scholars who accept the signal as strong evidence of pre-Columbian contact argue that the confluence of factors, the right geography, the right timing, the right population clustering, and the statistical robustness of the DNA results makes ancient contact the most parsimonious explanation available. The scholars who urge caution argue that the methods involved in admixture dating carry uncertainty ranges wide enough to allow for alternative explanations, that the source population on the American side remains imprecisely identified, and that a single genetic signal, however compelling, is stronger when it stands alongside other independent lines of evidence rather than alone. And here is where the stakes of this particular finding become clear. The South America signal is not merely an academic curiosity. It is one of the central pieces of evidence in a broader argument about what the pre-Columbian world actually looked like, whether ocean distances that seem vast on a modern map were genuine barriers to human connection, or simply challenges that some people in some periods had the skill and ambition to overcome. If the DNA holds up under continued scrutiny, and so far it has held up through multiple independent studies, then something happened in the Eastern Pacific before any European ship came close. The question of exactly what it was remains genuinely open, but the question of whether it happened at all looks increasingly settled. The genetics, compelling as it is, does not stand alone. Long before anyone extracted ancient DNA from a tooth or modelled admixture dating across chromosomes, there was a plant sitting in the middle of the Pacific that refused to make sense under the assumption of isolated, unconnected worlds. The sweet potato is a South American crop. Its wild ancestors grow in the Andes and the coastal regions of the continent. There is no natural habitat for it anywhere in Polynesia, no indigenous relative, no plausible mechanism by which it could have emerged independently on Pacific islands. Yet when European explorers arrived across Polynesia, they found the sweet potato already there, already cultivated, already embedded in agricultural systems and food cultures that had clearly been relying on it for a long time. That fact alone demanded an explanation, and the explanations available all led, eventually, back across the ocean. The timing of the sweet potato's presence in Polynesia is not a vague impression. Researchers have dated preserved sweet potato remains from archaeological sites across the region, and the results push the plant's presence back well before any European vessel reached these islands. Samples from the Cook Islands have been radiocarbon dated to around 1000 CE, possibly earlier. Sites in the Marquesas and in Hawaii show evidence of sweet potato cultivation that predates European contact by centuries. The most rigorous recent work, combining radiocarbon dating with careful attention to contamination and sample integrity, has consistently supported the conclusion that the sweet potato was growing in Polynesia somewhere between 1000 and 1400 CE. That window sits comfortably before the Spanish galleons and Dutch merchant ships that came much later. Whatever brought the sweet potato across the Pacific, it was not European trade. 3 mechanisms have been proposed to account for the crop's presence, and they differ enormously in what they imply about human behavior. The first possibility is that sweet potato tubers or seeds drifted across the ocean on their own, carried by currents and winds without any human assistance. The Pacific has powerful ocean circulation patterns, and the argument for natural dispersal has been made seriously by researchers who prefer not to invoke human agency without strong necessity. The problem is that sweet potato propagation works primarily through tubers and vine cuttings, rather than seeds built for long oceanic travel. The viability of tubers diminishes over weeks of saltwater exposure. Studies testing how long sweet potato material can survive oceanic conditions have produced skeptical results. While natural dispersal cannot be ruled out entirely, the evidence for it is weak enough that most botanists and archaeologists do not consider it a satisfying explanation for what the archaeological record shows. The second possibility is that contact was real but indirect. That plant material was passed along through chains of intermediate exchange rather than a single dramatic crossing. Under this scenario, neither Polynesians nor South Americans needed to complete a full transoceanic voyage. The crop could have moved island by island, people by people, through networks of contact that individually covered short distances but collectively bridged the Pacific. This model has some appeal because it reduces the burden on any single group to have accomplished something extraordinary. The difficulty is that there is no credible chain of intermediate islands between Polynesia and South America. The Eastern Pacific is not the Mediterranean. There are no stepping stones. Between the easternmost Polynesian islands and the South American coast lies an enormous stretch of open ocean with nothing in between. An indirect dispersal route does not work geographically. That leaves the third possibility, which is direct human transport. Either Polynesians who reached South America and brought the plant back, South Americans who reached Polynesia and brought it with them, or some combination of both. This is where the sweet potato evidence locks arms with the genetic findings described earlier. The same geography that makes natural or indirect dispersal implausible makes direct human voyaging the default explanation, and the DNA evidence suggesting pre-contact admixture between Polynesian and South American populations provides exactly the kind of human encounter during which crop exchange could plausibly have occurred. These 2 lines of evidence, one biological and one botanical, were developed largely independently, and yet they converge on the same conclusion in the same time period. That convergence is significant. The linguistic evidence adds another layer that is harder to dismiss. The word used for sweet potato in Polynesian languages across a wide geographic range is some version of the word kumara or kumala. Variations of this term appear in Hawaiian, Maori, Rapa Nui, Rarotongan, and other languages across the Polynesian family. What makes this linguistically striking is that a closely related word, kumar or kumara, appears in Quechua, the language spoken across much of the Andean region of South America. The resemblance between these terms has been noted since at least the 19th century, and it is not easily explained by coincidence. Language families separated by thousands of miles do not typically share vocabulary unless there has been contact between speakers. Words travel with things, and names for crops travel with the crops themselves. The Kumara connection suggests that whoever carried the sweet potato across the Pacific carried its name along with it, and that name came from South America. Some linguists have pushed back on this interpretation, arguing that the resemblance could be coincidental, or that reconstructing the direction of borrowing is more complicated than it appears. Others have pointed out that Quechua itself has a complex internal history, and that pinning down exactly which population used which term at which point requires historical linguistic work that remains incomplete. These are fair cautions. Linguistic evidence is rarely as clean as it looks from a distance. But the convergence of a similar agricultural word in two language families, with no other obvious historical connection, combined with the presence of the crop itself, is difficult to wave away without a compelling alternative account. What the sweet potato evidence does exceptionally well is demonstrate that the genetic findings are not an isolated anomaly. A DNA signal can be debated on technical grounds. Admixture dating carries uncertainty ranges. Reference population selection introduces complications. All of these methodological conversations are legitimate, and they matter. But the sweet potato cannot be wished away through debates about statistical modeling. It is a physical object. It was there, it was being eaten and cultivated across Polynesia centuries before any European ship could have brought it. Its presence demands a mechanism, and the only mechanisms that hold up geographically require human beings crossing the open ocean in both directions. At the same time, the sweet potato is not a substitute for the genetic evidence. It tells us that contact happened, or at least that something was transported. It does not tell us who made the crossing or how many times it occurred. It does not provide the kind of individual-level biological link that admixture analysis offers. A scenario in which a single canoe made a single crossing and returned with plant cuttings would be enough to explain the crop's presence, without requiring the kind of extended contact that might leave a stronger genomic signature. The sweet potato and the DNA each answer slightly different questions, and the value of having both is precisely that. Neither one has to carry the entire argument. Crop evidence establishes that something moved. Genetic evidence establishes that people mixed. Together, they describe a pre-Columbian Pacific that was connected in ways that the standard story of discovery has never adequately accounted for. The sweet potato is a strong case. It is physical, it is datable, and when you press on it from multiple directions, it holds. Not every piece of evidence for pre-Columbian contact has that kind of resilience. Some claims that once looked equally promising have collapsed under closer examination, and understanding why they collapsed is just as important as understanding why the sweet potato and the genetic findings have held up. Science is not only about accumulating evidence that supports a conclusion, it is also about watching what happens when that evidence gets tested. And the story of pre-Columbian chickens in South America is one of the most instructive examples of how fragile ancient contact arguments can be, when the methodology underneath them is not as solid as it first appears. The claim was straightforward, and when it arrived, seemed genuinely exciting. In 2007, a team of researchers published findings based on chicken bones excavated from an archaeological site on the coast of Chile called El Arenal. The bones were dated using radiocarbon methods to somewhere between 1304 and 1424 CE, a period that comfortably predates Columbus and the Spanish introduction of European livestock to the Americas. The researchers also extracted mitochondrial DNA from the bones and compared the sequences to chickens from across the world. What they found was that the Chilean chicken DNA showed the closest genetic match to chickens from Polynesia, not to the European breeds that would have arrived with Spanish colonizers. The conclusion seemed obvious, and the implication was significant. If Polynesian chickens had reached South America before European contact, it provided independent biological evidence for exactly the kind of transoceanic voyage that the genomic and botanical evidence was also suggesting. Polynesian sailors, the argument went, had not only brought people into contact with South America, they had brought their animals too. The paper received wide attention. It seemed to fit neatly into an emerging picture of a pre-Columbian Pacific that was far more connected than conventional history had assumed. The sweet potato moved east across the ocean. Genetic signals pointed to real population mixing. And now here were chicken bones in Chile, radiocarbon dated to before Columbus with Polynesian DNA. The convergence looked compelling. Then other researchers looked more carefully, and the confidence started to unravel. The reanalysis that followed raised two distinct sets of problems, and both mattered. The first concerned the radiocarbon dating. Radiocarbon dating works by measuring the decay of carbon-14 in organic material, but the method requires calibration curves that translate raw measurements into calendar years. Those calibration curves are not uniform across the world because organisms absorb carbon from their local environment, and in coastal regions where marine food sources contribute significantly to diet, the carbon signature can be skewed. This is called the marine reservoir effect. Animals or humans whose diet includes substantial marine input will produce radiocarbon dates that appear older than they actually are, because ocean carbon cycles on a different timescale than atmospheric carbon. The El Arenal chickens were from a coastal site. If they had eaten any marine material, their bones could carry a carbon signature that made them appear significantly older than they were. When researchers applied corrections for the potential marine reservoir effect, the date ranges shifted. Some estimates moved the bones into a period that could plausibly overlap with European contact rather than securely predating it. The apparently pre-Columbian date became considerably less certain. The second problem involved the DNA comparison. Ancient DNA work is technically demanding under the best circumstances, and the mitochondrial sequences recovered from the El Arenal bones were short and degraded. When the original team compared those sequences to global chicken populations, they found the closest match in Polynesian breeds. But the matching process depends on the reference database you're comparing against, and in 2007 that database had significant gaps. Later work with expanded reference populations found that the sequences from El Arenal were not uniquely Polynesian. European chicken breeds, particularly older or more diverse lineages, carried similar mitochondrial sequences. The genetic signature that had seemed to point specifically toward Polynesia turned out to be less diagnostic than it initially appeared. It was consistent with Polynesian chickens, but it was also consistent with other populations. When a piece of evidence is compatible with multiple explanations, it cannot support strong conclusions about any single one of them. By the time the critiques and rebuttals had run their course, what remained was a chicken bone that might have been pre-Columbian, or might not have been, with DNA that might have come from Polynesia or might have come from somewhere else. That is not nothing, but it is a very long way from the confident headline that first emerged. The researchers who published the original findings were not being careless or dishonest. They were working with the tools and reference databases available at the time, and they acknowledged uncertainties in the original paper. What the episode illustrates is how quickly apparent convergence can dissolve when each individual line of evidence is examined at the level of its underlying assumptions. This matters because the chicken story is sometimes cited alongside the sweet potato and the genetic admixture findings as though all three belong to the same category of evidence. They do not. The sweet potato argument has survived repeated scrutiny, including the application of increasingly rigorous radiocarbon protocols and expanding genetic databases for plant ancestry. The genomic admixture findings have been replicated across multiple independent studies using different methodologies and different reference populations. Each time researchers have tried to explain the signal away through technical means, the signal has persisted. The chicken bone evidence, by contrast, weakened under exactly this kind of pressure. That difference is not a minor detail. It is precisely the information you need to evaluate what the overall picture actually supports. The scientific disagreement around the chickens has never been fully resolved, which is its own kind of lesson. Some researchers still argue that the El Arenal evidence, even with its complications, is not definitively disproven. Others consider the case effectively closed. The literature contains pointed exchanges between teams with different readings of the same data, disagreements about how to apply marine reservoir corrections, and debates about which reference databases best represent global chicken diversity in the relevant time period. None of these arguments have produced a consensus verdict that satisfies everyone. In that sense, the chicken bones remain genuinely contested rather than simply wrong, and the distinction matters for how you characterize the state of knowledge. What the chicken case does not do is undermine the stronger evidence. A contested or weakened piece of evidence for a contact does not retroactively invalidate the genomic data or the sweet potato record. Arguments about pre-Columbian contact are sometimes dismissed wholesale when a particular supporting claim turns out to be shaky, as though the failure of one line of evidence proves the entire thesis wrong. That is not how evidence works. The case for pre-Columbian Polynesian contact with South America does not rest on the El Arenal chickens. It rests on admixture signals identified across multiple independent genomic studies, and on a crop whose presence in Polynesia cannot be explained without human transport. The chickens were interesting corroborating detail, and their story became more complicated. The core argument did not go with them. What the chickens do provide is a useful test of how the broader evidence should be handled. Not every intriguing result holds up. Ancient DNA degrades, calibration curves have regional quirks, reference databases grow and shift the interpretation of earlier findings, and what looks like a clean signal in one year becomes a contested data point in the next. Treating every promising result as confirmation and every complication as an attack on the entire field produces neither good science nor good history. The strength of the pre-Columbian contact argument lies precisely in the fact that its central claims have been subjected to this kind of pressure and survived it. The chicken bones remind you that not everything does. The chicken bone debate lands in a particular kind of ambiguity that is genuinely uncomfortable for anyone who wants clean answers. But not every pre-Columbian contact story sits in that uncertain territory. Some cases have moved well past debate and into the realm of documented historical fact, and the Norse presence in North America is the clearest example available. Understanding what made that case provable and what it still cannot tell us gives you a useful baseline for evaluating everything else. The site is called Lance Oaks Meadows, located on the northern tip of Newfoundland in Canada. When archaeologists began excavating there in the 1960s, what they found was unambiguous. Sod buildings constructed in a style consistent with Norse architecture from the Viking Age. Iron nails, a bronze cloak pin of Scandinavian design. A spindle whorl suggesting the presence of women. Butternuts, which do not grow in Newfoundland, indicating that the occupants had travelled further south into what is now New England or the Gulf of St. Lawrence region, and brought the nuts back. The radiocarbon dates from the site consistently cluster around 1000 CE, more than 4 centuries before Columbus crossed the Atlantic. There was no plausible mechanism by which all of this physical evidence could have appeared in Newfoundland other than Norse people actually being there. The site was designated a UNESCO World Heritage Site in 1978, and the scholarly consensus has never seriously wavered since. More recently, researchers refined the dating even further using a clever application of tree ring science. A solar storm in 993 CE produced a distinctive spike in atmospheric radiocarbon that became embedded in tree rings worldwide in exactly the same year. Wood from the Lance Oak Meadow structures contained that spike, and counting forward from it placed the cutting of those trees in 1021 CE. That is not an estimate or a range, that is a specific year derived from a known physical event embedded in the wood itself. The Norse were in North America in 1021 CE. That sentence no longer requires a hedge. What Lance Oaks Meadows proves for the broader history of Atlantic crossings is significant. It establishes that transoceanic voyages to the Americas happened before Columbus, that they were documented in written sources that turned out to be accurate, and that archaeological evidence of those voyages survives and can be dated with precision. The Norse sagas, describing encounters with a place the voyagers called Vinland, were not mythology. They were records of something real that should recalibrate how cautiously historians approach oral traditions and early written accounts of distant lands. The default skepticism that treats such sources as unreliable until physically confirmed remains appropriate, but Landsort Meadows demonstrates that the confirmation can eventually arrive. Now, here is where the DNA question becomes genuinely interesting and also genuinely frustrating. If the Norse were in North America for some period around 1000 CE, did they leave any detectable genetic trace in indigenous populations? The answer, based on current evidence, is almost certainly no, and understanding why that null result is not surprising tells you something important about the limits of genomic evidence as a historical tool. Landsort Meadows was a small settlement, probably occupied intermittently rather than continuously, and the Norse appear to have abandoned it relatively quickly. The sagas themselves describe conflicts with the indigenous people they encountered, whom they called Skraelings, and those conflicts are generally understood as a major reason the settlement did not persist. A small group of people occupying a site for perhaps a decade or two, with hostile rather than reproductive relations with the local population, leaves almost no opportunity for gene flow. For genetic admixture to show up in modern populations, you need sustained contact, intermarriage, and descendants who survived and reproduced across generations in sufficient numbers to carry detectable proportions of foreign ancestry forward through time. A brief Norse outpost that was abandoned and never returned to does not create those conditions. This is a fundamental constraint on what ancient DNA evidence can detect. It finds contacts that were reproductively successful, meaning contacts that resulted in children who had children across multiple generations in large enough numbers to leave a statistical signal. It does not find visits. It does not find trade expeditions that lasted a season. It does not find settlements that were established, contested, and abandoned within the span of a human generation. The Norse in Newfoundland fall into that last category. Their presence is archaeologically certain and genetically invisible, and those two facts are completely compatible with each other. There has been one genetic study that attracted attention for a different Norse angle. Research into Icelandic population genetics identified a mitochondrial lineage in a small number of Icelandic families that appeared to have Native American origins, with a mutation pattern suggesting it entered the Icelandic gene pool sometime around 1000 CE. The interpretation offered was that a Native American woman had travelled or been brought to Iceland during the Norse period, possibly during or after the Vinland voyages. If correct, this would represent detectable genetic evidence of contact flowing in the other direction, from the Americas into Europe, rather than from Europe into the Americas. The finding has been discussed cautiously, with researchers acknowledging the difficulty of ruling out more recent explanations, but it has not been definitively disproven either. It remains a suggestive data point rather than a confirmed conclusion, which places it in a different evidentiary category than the archaeological findings at Landsort Meadows. Comparing the Norse case to the Pacific contact evidence reveals something structurally important about how different types of proof work. The Norse presence in North America is proven archaeologically beyond reasonable doubt and is genetically invisible. The Polynesian contact with South America is proven genomically across multiple independent studies and is archaeologically ambiguous, with the sweet potato providing strong botanical support, but no excavated settlement site equivalent to Landsort Meadows yet identified. These are two very different evidentiary profiles reaching the same basic conclusion, which is that pre-Columbian contact across major ocean crossings happened. Neither case depends on the type of evidence that made the other case convincing. That matters because critics of the Pacific evidence sometimes point to the lack of a smoking gun archaeological site as a reason for doubt. But the Norse case shows that archaeology and genetics do not always preserve contact in the same way, or even at all. A contact event that was brief and contentious leaves physical remains without genetic traces. A contact event that involved meaningful population mixing may leave genetic traces without obvious physical remains, particularly if the mixing happened at sea or in locations not yet excavated. Demanding that every contact leave both kinds of evidence before it counts as real is a methodological standard that the Norse case itself cannot fully meet. What Landsauk's Meadows ultimately provides is not just confirmation of Viking Age Atlantic crossings. It provides a well-documented test case for how pre-Columbian contacts work in the historical and scientific record. Some leave wood and iron nails, some leave signatures in DNA, some leave crop plants distributed across thousands of miles of ocean. What you cannot assume is that absence of one type of evidence is absence of the event itself. The Norse were there, the genetics are silent. Both of those things are true, and neither one cancels out the other. The Norse case makes one thing unmistakably clear. Ocean crossings and settled coastlines were not the only pathways through which people, ideas, and biological material moved across vast distances before recorded history. There is a second model of early contact that gets far less dramatic attention than open ocean voyages, and it deserves the same careful examination. It runs along the top of the world, through the Arctic, and the people who maintained it were among the most mobile and geographically sophisticated on Earth. The Inuit and their predecessors did not think of the Arctic as a barrier, they thought of it as a highway. The Thule culture, the ancestral population from which modern Inuit descend, expanded out of Alaska roughly 1,000 years ago and moved eastward across the Canadian Arctic and into Greenland with extraordinary speed by any archaeological measure. Within a few centuries, Thule people had traversed the entire width of Arctic North America, a distance comparable to crossing a continent. They did this not by accident or as a slow generational drift, but through deliberate, organized movement enabled by sophisticated technology, including large umiaks capable of open water travel, dog sleds for overland and sea ice routes, and the knowledge to navigate environments that would kill an unprepared traveler within hours. These were not isolated communities scraping out survival in a hostile landscape. They were connected societies operating across enormous territories with maintained communication between distant groups. The genetic record of this expansion is detailed and legible. Ancient DNA studies of Arctic populations show clear evidence of population structure, movement and replacement across the far north. Earlier populations, including the Dorset culture people who occupied much of the Arctic before Thule expansion, were largely replaced rather than absorbed, though recent analysis has found subtle traces of Dorset ancestry in some later populations, suggesting at least limited genetic mixing at the boundaries of these expanding and contracting groups. What the genomics reveal overall is a pattern of dynamic population movement rather than static isolation, with different groups expanding, contracting, and occasionally exchanging genes across the top of the continent over thousands of years. Now place Greenland into this picture. When the Norse established settlements in Greenland around 985 CE, they were moving into territory that was already occupied or had recently been occupied, and that would later be fully occupied by Thule Inuit expanding from the west. The Norse Greenland settlements survived for roughly 4.5 centuries before disappearing, and the question of what happened to them has generated significant historical and scientific debate. One persistent question is whether there was meaningful contact, including genetic contact between Norse Greenland settlers and the Inuit populations they shared that island with for at least part of that period. The Norse written sources mention encounters with Skraelings in Greenland as well as in Vinland, and archaeological sites in Greenland have produced objects of Norse origin in Inuit contexts, including wooden carvings that appear to depict people in European-style clothing, chainmail fragments, and other manufactured goods. The material exchange was real. Whether it ever extended to the biological is harder to establish, though genetic studies of Greenlandic Inuit populations have not yet produced the kind of strong admixture signal that would settle the question definitively. It remains an area of active investigation rather than a closed case. What makes the Arctic corridor conceptually important is not just the question of Norse-Inuit interaction, but the broader principle it illustrates about how contact can work. Open ocean voyages like those of the Polynesians involve populations deliberately crossing large stretches of water in a single expedition, arriving somewhere distant, and potentially leaving a direct genetic or material signature at the destination. The Arctic model works differently. It involves chains of neighbouring populations, maintaining exchange networks across contiguous territory, passing goods, people and genes through a series of short-range contacts that accumulate over time into effectively long-range transmission. You do not need anyone to sail from Alaska to Greenland in a single voyage if there are populations at regular intervals across the Arctic who trade with their neighbours, sometimes intermarry, and occasionally absorb individuals from adjacent groups. Information, objects, and ancestry can travel enormous distances through that kind of network, without any single person ever making the full journey. This indirect contact model has profound implications for how we think about pre-Columbian connectivity. Genetic admixture studies do not always distinguish cleanly between a single long-distance contact event and a gradual diffusion of ancestry through a chain of intermediate populations. Both processes can produce detectable signals in modern or ancient genomes, but they leave different signatures if examined closely enough. Researchers working on Arctic population genetics have increasingly emphasized the importance of looking not just at the endpoints of potential contact, but at the intermediate populations that might have served as conduits. If a particular genetic variant shows up in both a Siberian population and an Alaskan one, the question is not simply whether those two populations were ever in direct contact, but whether the variant moved through a network of exchanges that included populations now extinct or unsampled, whose intermediate role has been erased from the visible record. The Bering Strait region is the most studied example of this kind of corridor contact. Genetic exchanges between Siberia and Alaska have been documented across multiple time periods, with different lineages moving in different directions at different times. Some of these exchanges involved large-scale population movements. Others appear to have been small-scale and recurring, consistent with the kind of sustained trade and social interaction that historical and ethnographic records describe between peoples on both sides of the strait. The strait itself is narrow enough that on clear days you can see one shore from the other, and indigenous peoples on both sides maintained active contacts documented in both oral traditions and early European accounts. Treating that strait as a genetic and cultural boundary rather than a connection point would be a significant distortion of the actual history. The Atlantic side of the Arctic presents a different but equally interesting picture. The North Atlantic, while treacherous, is not an unbroken expanse of open ocean. It is dotted with islands from the Faroes to Iceland to Greenland that served as stepping stones for Norse expansion, and that also place Greenland within a connected Atlantic world rather than an isolated outpost. Genetic studies of Icelandic and Greenlandic populations have traced the layering of different ancestries brought by different migration events, and the picture that emerges is one of cumulative movement rather than a single founding voyage. Each population that settled these North Atlantic islands brought ancestry from somewhere else, and sometimes mixed with populations already there, creating genomic palimpsests where multiple contact events are stacked on top of each other. Placing Arctic routes alongside open ocean Pacific voyages as parallel models of early contact is not just an academic exercise in taxonomy. It changes what questions you ask and where you look for evidence. If you only think about contact in terms of deliberate long-distance voyages, you will look for arrival sites, founding populations, and dramatic genetic breaks. If you also think about contact in terms of sustained exchange networks operating through chains of neighbouring peoples, you will look for gradual climbs in genetic variation, shared material culture at multiple sites across a region, and the kind of slow diffusion that leaves a different archaeological and genomic signature. The Arctic evidence suggests that both models were operating simultaneously in the pre-Columbian world, and that the full story of early human connectivity requires holding both in view at once. The people who moved through the Arctic were not marginal actors in human history, doing something simpler or less consequential than the ocean navigators of the Pacific. They were maintaining complex societies across some of the most demanding environments on Earth, building and sustaining networks that stretched across thousands of miles, and doing so with a degree of geographic knowledge and technological sophistication that made them, in their own domain, every bit as capable as the sailors who crossed open ocean to the south. The Arctic routes and Pacific Ocean crossings represent two different models of how people moved across vast distances before the written historical record catches up with them. But if you want a single case study that cuts through every theoretical debate and lands with undeniable clarity, you look south. You look at Madagascar. Madagascar sits in the Indian Ocean off the eastern coast of Africa. Geographically, that placement makes a simple prediction. The island should have been settled by Africans. People from the nearby continent, separated from Madagascar by a few hundred miles of open water, should have been its first inhabitants, and their descendants should make up the majority of the population today. That is the obvious story. It is not what the DNA shows. The Malagasy people, the diverse population of Madagascar, carry ancestry from two distinct sources. One of those sources is Africa. The Bantu-speaking populations of East Africa contributed a real and significant genetic component to the Malagasy gene pool, and the evidence for that contribution is clear in both ancient and modern genomic data. But the other source is not from anywhere nearby. It is from Southeast Asia, specifically from populations ancestrally related to the people of Borneo, situated roughly 8,000 kilometers to the east across the Indian Ocean. That Southeast Asian genetic signal is not a trace. It is not an ambiguous statistical ghost. It is a founding component of Malagasy ancestry, present in substantial proportions in populations across the island, and it is accompanied by cultural and linguistic evidence so consistent that every line of inquiry points in the same direction. The Malagasy language belongs to the Austronesian family. That family of languages includes the languages of maritime Southeast Asia, the Pacific Islands, and the scattered island populations across a vast portion of the globe. The closest relative of Malagasy within that family is a language spoken in Borneo, a connection so specific that linguists identified it before genetics ever entered the conversation. The vocabulary, the grammar, the structural features of Malagasy all point to an origin among Austronesian-speaking peoples of island Southeast Asia, not among the Bantu-speaking peoples of the African mainland. Language does not usually survive intact if a founding population is absorbed into a much larger neighbouring group. The preservation of an Austronesian language in Madagascar means the Southeast Asian founders were not just visitors or traders who left a few descendants behind. They were settlers who established a community substantial enough to transmit their language across generations and across the centuries, until the island was eventually contacted by and partially merged with African populations from the west. Genetic studies have refined this picture considerably in recent years. Analysis of both modern Malagasy genomes and, where available, ancient skeletal material has confirmed the dual African and Southeast Asian ancestry, and researchers have worked to estimate when each component arrived and in what proportions. The Southeast Asian ancestry appears to date to roughly 1,200 years ago, though estimates vary depending on methodology, and the African genetic contribution reflects subsequent contacts and migrations from the East African coast. Some populations within Madagascar show higher proportions of one ancestry than the other, reflecting the island's internal history of different groups settling different regions and mixing in different degrees. But across the island as a whole, the Southeast Asian genetic signal is unmistakable and substantial, and it cannot be explained by anything other than a founding migration from that region. The implications for Indian Ocean navigation are significant. The Austronesian-speaking peoples who settled Madagascar were not crossing a short stretch of coastal water. They were crossing the full width of the Indian Ocean, one of the world's major ocean basins, characterized by powerful monsoon systems, long swells, and no intermediate islands to serve as staging posts for a gradual island-hopping route. The voyage from Borneo or the broader maritime Southeast Asian region to Madagascar requires either traversing the Indian Ocean directly or following a longer route through South and Southeast Asian coastal waters before striking west across open ocean. Either way, it is a journey of thousands of kilometers involving blue water sailing capability, navigational knowledge, and the logistical capacity to survive at sea long enough to make landfall. The technology that made this possible was the outrigger canoe, the same basic vessel type that Austronesian peoples used to colonize the Pacific. The double outrigger or single outrigger canoe designs developed in maritime Southeast Asia gave sailors platforms that were both fast and stable in open ocean conditions, capable of sailing against the wind as well as with it, and durable enough for extended voyages. The monsoon wind systems of the Indian Ocean, which reverse direction seasonally, would have provided predictable sailing windows in both directions, allowing not just a one-way migration, but the possibility of sustained contact between regions. There is archaeological and historical evidence for Austronesian maritime trade networks operating across the Indian Ocean, well before the first millennium CE, and the settlement of Madagascar fits within that broader pattern of Indian Ocean connectivity rather than standing as an isolated anomaly. This is the point where Madagascar becomes more than just a fascinating piece of its own regional history. It becomes a comparative case for how we evaluate other seemingly implausible maritime scenarios. Before the genomic evidence was assembled, before the linguistic relationship to Borneo languages was fully worked out, the idea of prehistoric sailors from Southeast Asia founding a population on the eastern shore of Africa would have seemed like speculation. The distances involved are too large. The technology seems too basic. There are no obvious intermediate way stations. The story sounds too extraordinary to be taken seriously as history rather than mythology. And yet it happened. The DNA proves it happened. The language proves it happened. The convergence of independent lines of evidence from genetics, linguistics, and archaeology creates a case that has no credible alternative explanation. What that means for every other seemingly implausible ancient maritime scenario is that the prior probability of such events needs to be revised. When geneticists find an unexpected ancestry signal in a population that should not, on geographic logic alone, carry it, the response cannot simply be to dismiss it as a statistical artifact or explain it away through some simpler mechanism. Madagascar teaches us that Austronesian sailors moved 8,000 kilometers across open ocean and founded a civilization on the far side of an ocean basin, and they did it over 1,000 years ago without the navigation instruments, communication technology, or institutional support that we associate with serious ocean exploration. If that happened, then other extraordinary voyages need to be taken seriously on their evidence, not dismissed on their surface improbability. The Indian Ocean world of the first millennium CE was more connected than most historical narratives acknowledge. Arab, Indian, Southeast Asian, and East African maritime traders were operating across that basin, carrying goods, people, languages, and genes in multiple directions along routes that predate the era of European exploration by centuries. Madagascar sits at the western edge of that world, settled by people from its eastern edge, and its existence as a populated island is a permanent reminder that the ocean itself was never the obstacle it appears to be on a flat map. To the people who understood its winds and currents, who built the right vessels and learned to read the sky and sea, it was a highway rather than a barrier. That reframing from ocean as obstacle to ocean as pathway is the conceptual shift that the Madagascar case crystallizes better than almost any other single example. The astonishing voyage did happen. The genetic record confirms it. And once you accept that it happened there, you have to take seriously the evidence that similar things happened elsewhere, in the Pacific, in the Arctic, and in places where the signals are fainter, but the logic of human capability points in the same direction. Madagascar gave us a case where the genetic signal is loud. The Southeast Asian ancestry in the Malagasy population is not subtle. It does not require specialized statistical methods to detect or careful squinting at marginal probability distributions. It is right there in the data, abundant and consistent, accompanied by linguistic evidence that removes any remaining ambiguity. But Madagascar is in an important sense the easy version of this problem. Most ancient contacts did not leave signals that clear. Most of the time, when two populations met briefly at the edge of the world 1,000 years ago, what they left behind in the genome is something far harder to read. A whisper rather than a shout. And understanding why that whisper exists, and why it is sometimes inaudible entirely, is essential to understanding what DNA can and cannot tell us about the full history of human movement across the planet. Start with the basic arithmetic of genetic inheritance. When two populations make contact, the amount of shared ancestry that enters the gene pool of either group depends directly on how many people were involved in that contact and what happened to them afterward. A large founding migration, like the one that established the Malagasy population, introduces substantial genetic material from the source population, and that material persists across generations in proportions that remain detectable, even after centuries of mixing with other groups. But a small contact event, a single canoe of sailors who arrived, traded, had children with a handful of local people, and then left or were absorbed, introduces a much smaller genetic contribution. That contribution gets diluted with every passing generation as descendants of the contact intermarry with the surrounding population. After enough generations, the original signal may fall below the threshold of reliable statistical detection, becoming what researchers call a ghost in the genome. Something happened. The mathematics of population genetics suggests something must have happened, but the direct evidence of it has been washed out by time and demography. This is not a hypothetical problem. It describes the actual situation that geneticists face when trying to reconstruct ancient contacts in many parts of the world. The statistical tools used to detect admixture events, to identify moments when two previously separated populations exchanged genetic material, all have sensitivity limits. Below a certain threshold of admixture proportion, the signal becomes indistinguishable from noise, from the random variation in allele frequencies that accumulates in any finite population over time. Ancient contacts that involved small numbers of people, brief duration or limited subsequent reproduction can fall below that threshold entirely, leaving no recoverable trace in the genomes of living descendants. The problem is compounded by what happens when later migrations arrive. If a population that carries a trace of ancient contact is subsequently overwhelmed by a larger migration from a different source, the newer genetic material can dilute or entirely mask the earlier signal. Think of it as layers of paint on a canvas. The original layer is still there physically, but you cannot see it anymore through everything applied on top. Researchers working with ancient DNA from skeletal remains sometimes find ancestry patterns in early individuals that have completely disappeared from the modern population of the same region, replaced by the genetic signature of later arrivals. This means that limiting the search to modern genomes systematically underestimates the complexity of ancient contact history. The living population is not always the most informative witness to what happened in its own past. Ghost populations take this challenge one step further. In some cases, geneticists identify statistical evidence for ancestry in a sampled population that cannot be matched to any known source group, living or ancient. The modelling requires an unknown ancestral component, one that must have existed based on the mathematics of allele frequency distributions, but whose actual carriers have never been sampled because no skeletal remains from that group have been found and sequenced, or because the group in question left no surviving descendants. These are called ghost populations, and they appear with surprising frequency in sophisticated genomic analysis of human prehistory. Their existence can only be inferred indirectly through the statistical signatures they left in the genomes of populations they once mixed with. We know something was there. We cannot directly observe what it was. The practical implication for ancient maritime contact is significant. When researchers look for evidence of pre-Columbian contact between the Pacific and the Americas, or between Atlantic populations and the New World, they are often searching for precisely this kind of ghost signal. The contact hypotheses being tested typically involve small numbers of people, episodic rather than continuous interaction, and time depths great enough that subsequent demographic changes would have had ample opportunity to dilute whatever genetic trace was introduced. Finding a clear signal under those conditions is genuinely difficult, which means failing to find a clear signal does not straightforwardly mean the contact did not occur. Absence of detectable genetic evidence and absence of actual contact are not the same thing. The distinction matters enormously for how we interpret negative results. Scientists working in this field navigate this problem through several approaches. One is to use ancient DNA wherever possible, sampling individuals who lived closer in time to the hypothesized contact event before later migrations had a chance to overwrite the earlier signal. Another is to combine genetic evidence with non-genetic lines of evidence, looking for convergence across archaeology, linguistics, biological anthropology, and historical records. A single ambiguous genomic signal is weak evidence. The same signal accompanied by an archaeological artifact of foreign origin, a linguistic borrowing with no obvious transmission route, or a plant species appearing in a region before any known introduction event becomes considerably more compelling. The methodological ideal is triangulation across independent data types, each with its own error structure and its own blind spots, such that what all of them agree on is more likely to be real than what any one of them claims alone. There is also the honest acknowledgement that some contacts will simply never be recoverable through any currently available method. If a handful of sailors reached a coast, left no objects behind, fathered no children with the local population, and returned home or died without founding a lineage, there is nothing for either the archaeologist or the geneticist to find. History is not obligated to leave traces legible to modern science. The contacts that have been documented through DNA are the ones that crossed certain demographic and temporal thresholds. Below those thresholds, the record goes dark, not because nothing happened, but because what happened was too small or too long ago or too thoroughly overwritten to survive in recoverable form. What all of this means for the broader project of reconstructing ancient maritime history is that genetic evidence is a powerful but bounded tool. It can confirm contacts that other evidence suggests, rule out some proposed explanations, and occasionally reveal connections that were entirely unsuspected. But it cannot provide a complete inventory of every crossing, every landfall, every brief exchange between peoples separated by an ocean. The contacts that left clear signals, like the ones that established Polynesian ancestry traces in South American populations or founded the Malagasy people, represent a recoverable subset of ancient human interaction, not its totality. The full picture includes an unknown number of ghost contacts, faint signals degraded beyond recognition, and events that left no biological trace at all. This probabilistic nature of the evidence is not a weakness of the field. It is an accurate description of what history actually looks like when you try to reconstruct it from physical remains after 1,000 years. The scientists working with these tools are not claiming certainty they do not have. They are building probabilistic cases, assigning confidence levels, acknowledging uncertainty, and revising their conclusions as new data arrives. That is how the method is supposed to work, and it is precisely why the cases where the evidence does converge clearly, where multiple independent signals all point in the same direction, carry the weight they do. The picture we have built across this story has not come from any single line of evidence. It has come from the collision of several, and that collision is often uncomfortable. Genetics tells one part of the story, archaeology tells another. Linguistics adds its own layer. botany weighs in with plants that should not be where they are, and oral traditions carry memory of events that written records never captured. Each of these disciplines has its own methods, its own standards of proof, its own blind spots. When they agree, the case becomes powerful. When they disagree, which happens more often than textbooks suggest, the disagreement itself becomes scientifically productive, forcing researchers to ask harder questions than any single field would have generated on its own. Consider what each type of evidence actually measures. Genetics captures biological relatedness across generations. It tells you that people who share ancestry once shared geography at some point in the past, though pinning down exactly when and where requires careful modelling with real uncertainty attached. Archaeology captures material culture at specific locations and specific moments, but only what was preserved, only what was excavated, and only what was correctly interpreted. Linguistics tracks the branching and borrowing of languages, which can reveal contact between populations even when no other record survives. But the dating of linguistic divergence carries its own error bars and its own contested methodologies. Botany, particularly the study of cultivated plant distributions, can identify movement of crops across geographic barriers that should have stopped them, but plant introduction events are notoriously hard to date precisely, and multiple introduction routes are often possible. Oral tradition carries cultural memory in some cases across extraordinary spans of time, but it is also subject to reinterpretation, embellishment, and the gradual conflation of separate events into single narratives. None of these sources is purely reliable on its own. None of them is purely unreliable either. The real analytical work happens when you try to build a case that holds across all of them simultaneously, and that is where the difficulty lives. The sweet potato is one of the clearest examples of how these lines of evidence can both converge and diverge in the same case. Genetically, the sweet potato is South American in origin, full stop. Botanically, it was present across Polynesia before European contact. Those two facts together constitute very strong evidence that someone carried it across the Pacific. Genetic analysis of pre-European sweet potato samples from Polynesian archaeological sites points toward a South American source population that diverged from the cultivated lineage around 1,000 years ago, which aligns with the period when Polynesian expansion was reaching its eastern limits and when Polynesian DNA begins appearing in populations on the South American coast. So far, the evidence triangulates, but when you try to pin down the mechanism, the direction of travel, whether Polynesians reached South America or South Americans reached Polynesia, or both, the lines of evidence start to pull in slightly different directions. The genetic signal in South American coastal populations suggests the contact was real and involved actual people, not just goods. But the archaeological record in South America shows no Polynesian material culture, no artifacts, no sites that unambiguously document Polynesian presence on the continent. Linguistically, there is limited evidence of borrowing in either direction. The case is strong enough to be convincing as a whole, while remaining genuinely unresolved in its details. Dating mismatches create some of the most persistent disputes in this field. When a genetic analysis estimates a contact event at 800 years ago and an archaeological site places the relevant material at 600 years ago, the gap may or may not be significant depending on the confidence intervals involved. Population genetic dating methods produce ranges, not precise years. Radiocarbon dating produces ranges with their own calibration uncertainties. When those ranges overlap, the apparent discrepancy can dissolve. When they do not overlap, something is genuinely wrong with at least one of the interpretations, and figuring out which requires revisiting the assumptions behind both. The chicken bone controversy demonstrated exactly this dynamic. Initial radiocarbon dates on pre-Columbian chicken bones from a Chilean site seemed to support Polynesian contact. Subsequent analysis suggested the dates were contaminated and the bones were post-European. The genetic analysis of the bones further complicated matters, with different research teams reaching different conclusions about whether the DNA matched Polynesian or European chicken lineages. The evidence did not converge, it collided, and that collision produced years of productive disagreement that is still not fully resolved. The standard of proof required to claim ancient transoceanic contact is genuinely high, and it should be. These claims overturn established narratives, sometimes dramatically. The scientific community is right to demand convergent evidence from multiple independent sources before accepting them. A single anomalous radiocarbon date is not enough. A single genetic signal that could be explained by population structure or methodological artifact is not enough. A single artifact of uncertain provenance is not enough. What builds a convincing case is when the genetic signal, the archaeological evidence, the linguistic data, and the botanical record all point toward the same conclusion independently, using methods that have different failure modes so that the same error cannot simultaneously explain all of them. The Norse in North America illustrate what that convergence looks like when it succeeds. The saga literature described settlements in a place called Vinland. Skeptics treated this as legend for a long time. Then the archaeological site at Lansorks Meadows in Newfoundland provided physical confirmation that Norse people had built structures there around 1,000 years ago. The genetic evidence followed, identifying European ancestry in indigenous North American populations in patterns consistent with brief Norse contact. The linguistic record is thinner, but the archaeological and genetic convergence was enough. No single piece of that evidence was decisive on its own. Together they were. The interdisciplinary cases built around Polynesian expansion demonstrate something equally important. The process is iterative. Evidence does not arrive all at once, neatly packaged. It accumulates over decades, with each new data set either reinforcing or complicating what came before. The geneticists need the archaeologists to tell them where to look for ancient samples. The archaeologists need the geneticists to tell them whether the people they excavated were who they appeared to be, and where they came from. The botanists need both to understand the human behavior that moved plants across oceans. The linguists provide a check on all of them, because language change follows its own logic that biological and material evidence cannot fully replicate. When these disciplines work in isolation, they each tend to overstate their conclusions. When they work in genuine dialogue, acknowledging each other's uncertainties, the result is a more accurate and more honest picture of what actually happened. What this means practically is that the strongest claims in this entire area of research are not the ones supported by the most dramatic single finding. They are the ones that have survived the most interdisciplinary scrutiny. A genetic result that archaeologists cannot yet account for is a hypothesis, not a conclusion. A hypothesis that has since been supported by botanical evidence, refined by ancient DNA from excavated remains, and found consistent with linguistic analysis is something considerably more solid. The gap between those two states is where most of this science currently lives, building the case, testing it against new evidence, revising when revision is required. That is not a weakness of the research. It is what rigorous inquiry into a genuinely difficult question actually looks like from the inside. The evidence has been accumulating for decades now, and the pattern it traces is unmistakable. Ancient peoples crossed oceans, reached continents, and made contact with populations that no conventional history recorded them meeting. The genetics confirm it, the botany supports it, the archaeology piece by piece is catching up. So a reasonable question hangs over all of it. If the evidence is this substantial, why did the old discovery stories last as long as they did? Why did the narrative of European explorers arriving at places no one had visited hold together for centuries, even when indigenous peoples had clearly been living there all along, and even when the physical and biological record pointed toward far older and more complex patterns of human movement? The answer is not primarily about evidence. It is about who controlled the institutions that evaluated evidence, who got to write the histories, and whose knowledge was treated as history at all. The concept of discovery, as it developed in the European legal and political tradition, was never really about finding a place that no human being had set foot in. It was about formal European recognition. When Spanish, Portuguese, English and French explorers planted flags and filed claims, they were operating within a doctrine that held European Christian sovereigns as the legitimate arbiters of territorial ownership. If a place was not already held by a Christian monarch, it was legally available for claiming, regardless of how long people had been living there. This doctrine, articulated in papal bulls and colonial charters, explicitly framed indigenous presence as legally irrelevant to the question of discovery. So the category was never an empirical one to begin with. It was a legal instrument designed to legitimize territorial acquisition, and it was written by the people doing the acquiring. That framing shaped historiography for centuries. European written records became the default primary sources for historical knowledge about these regions, not because they were the most accurate or the most complete, but because they were produced by the institutions that controlled universities, archives, publishing houses, and the training of historians. A Spanish chronicle describing first contact with a coastline was treated as historical documentation, an oral tradition held by the people who had been living on that coastline for generations was treated as legend, mythology, or at best an unreliable folk memory requiring corroboration from a written European source before it could be taken seriously. The asymmetry was not accidental. It was built into the epistemological infrastructure of colonial scholarship, where objectivity was coded as European literacy and subjectivity was assigned to everyone else. Indigenous navigation knowledge suffered a particularly sharp version of this dismissal. Polynesian wayfinding is a technical discipline of extraordinary sophistication. It involves reading star positions, ocean swells, cloud formations, bird behavior, water temperature, and bioluminescence across thousands of miles of open ocean. Generations of master navigators trained for years to internalize knowledge that allowed them to cross the Pacific with deliberate precision. When European scholars encountered accounts of this navigation, many of them reached for explanations that minimized the intentionality involved. Perhaps the voyages were accidental, perhaps people were blown off course, perhaps the distances involved were shorter than they appeared. The idea that Pacific Islanders had developed a navigational system more suited to open ocean travel than anything in the European repertoire at the same the me same dynamic played out across the Indian Ocean, the Arctic, and the coastlines of the Americas. When Inuit oral traditions described contacts and movements across enormous Arctic distances, or when West African oral histories referenced transoceanic connections, the burden of proof placed on these accounts was categorically different from the burden placed on European documentary sources, which had their own problems with accuracy, bias, and deliberate falsification. The standards were not neutral. They reflected the power relationships of the institutions setting them. This is the context into which DNA evidence has arrived, and it has arrived with a kind of authority that is difficult to dismiss on the same grounds. Ancient genomics does not rely on European archives. It does not depend on who wrote down what after which conquest. A genetic signal in a population is not a colonial document. It does not have an author with political motivations. It has error bars and methodological assumptions that can be interrogated, but it cannot be explained away by pointing to the biases of a Spanish chronicler or the convenient interests of an imperial court. When the DNA of a coastal South American population contains a clear Polynesian ancestry component dating to before European contact, that result stands outside the documentary tradition that shaped colonial historiography. It requires a different kind of explanation, and the most straightforward one is that people made contact. This is why DNA evidence has been so useful for historical revision, not because genetics is the only valid form of evidence, but because it enters the conversation from a direction that bypasses the institutional gatekeeping that marginalized indigenous accounts for so long. It does not prove that every oral tradition was perfectly preserved, or that every claim of ancient contact is valid. What it does is establish that the default framework, in which complex transoceanic contact was essentially a European innovation, is empirically wrong. And once that framework fails, the accounts and traditions that were dismissed within it deserve serious reconsideration. Revising discovery narratives along these lines does not require accepting every fringe claim that has accumulated around the edges of this research. The history of contact studies includes serious scholarship, and it also includes a substantial volume of speculation, motivated reasoning, and outright fabrication. Claims of ancient Egyptian contact with the Americas or Roman shipwrecks off the coast of Brazil, or the lost continents beloved by a certain genre of popular history, do not gain credibility simply because the old discovery narratives have been shown to be wrong. The evidentiary standards that genetics, archaeology, and linguistics apply are the same regardless of the cultural or political implications of the conclusion. A claim needs to meet those standards to be accepted, and many fringe claims conspicuously do not. What the evidence does support is a more crowded, more mobile, and more interconnected pre-Columbian world than the discovery framework allowed. Polynesian sailors reached South America and left a biological trace. Norse settlers built structures in North America 5 centuries before Columbus sailed. Austronesian-speaking people colonized Madagascar from Southeast Asia across thousands of miles of open ocean. The Inuit moved through Arctic corridors that connected continents. These are not fringe claims. They are conclusions supported by converging evidence from multiple independent disciplines, and they have survived rigorous scrutiny. The discovery stories lasted because they served the political purposes of the institutions that wrote and maintained history. They encoded legal claims to territory, organized the world into civilized discoverers and undiscovered peoples, and marginalized knowledge systems that complicated the picture. They lasted in academic settings because the methodological standards applied to non-European sources were designed in ways that made those sources easier to dismiss. They are now eroding not because the politics have simply shifted, though they have, but because the physical evidence embedded in human genomes, in ancient bones, in the genetic signatures of cultivated plants, does not answer to the same institutional pressures that shaped the written record. The DNA existed before the discovery doctrine was invented. It has been waiting to be read. Not everything in this field is settled. The previous section traced how DNA evidence has dismantled some of the most durable myths in conventional history, and that dismantling is real. But it would be a mistake to walk away from this research thinking the picture is now clear and the major questions have been answered. Some of the most important questions are still open, and in a few cases, the available evidence points in directions that actively contradict each other. Distinguishing between what is firmly established and what is still genuinely uncertain is not a minor housekeeping exercise. It is the difference between science and storytelling. Start with what can reasonably be called strong cases. The Polynesian contact with South America before European arrival is among the most robust findings in recent archaeogenomics. The genetic signal appears in multiple independent population samples. The dating is internally consistent, and the sweet potato evidence from botany reinforces the genomic conclusion from a completely different evidentiary direction. The Norse presence at Lands-Ork Meadows is established by direct archaeology, supported by radiocarbon dating and material culture analysis, and consistent with the Vinland sagas in their broad outlines. The Austronesian settlement of Madagascar from Southeast Asia is confirmed by genetics, linguistics, and material culture with high confidence. These conclusions are not beyond revision, nothing in science is, but they have survived serious scrutiny from multiple angles. Then there are the plausible but unproven cases, and this category is larger and messier. The question of whether Polynesian contact with South America was a single event or multiple events remains unresolved. The question of which specific Polynesian island group made the contact at what point on the South American coast and how sustained the interaction was, these are all genuinely open. The chicken bone evidence, which seemed at one point to offer a direct biological confirmation of Polynesian contact with pre-Columbian Chile, has not held up cleanly. Some analyses support a pre-Columbian Pacific origin for those chickens. Others suggest contamination or post-contact introduction. The chicken remains a contested clue rather than a settled piece of evidence. In the Atlantic, the picture outside of Norse Newfoundland is considerably murkier. There is an enduring question about whether significant pre-Columbian contact occurred between West Africa and the Americas. Some researchers have pointed to physical and cultural similarities that they argue are difficult to explain through convergence alone, and a small number of genetic studies have identified signals that might indicate pre-Columbian African-American contact. But the genetic evidence here is far weaker than in the Pacific case, and it is substantially harder to disentangle from the documented massive forced movement of African people during the Atlantic slave trade, which began shortly after Columbus and introduced African ancestry across the Americas at enormous scale. Separating a hypothetical pre-Columbian signal from that overwhelming post-Columbian one requires a level of ancient DNA precision that current sampling has not yet achieved. That brings up one of the biggest structural problems in this field. Ancient DNA sampling is uneven in ways that systematically shape which questions can even be asked. The ancient genomic record for the Pacific is richer than it was a decade ago, but still sparse. For large parts of South America, the ancient DNA record is fragmentary, partly because tropical and subtropical climates degrade biological material quickly, and partly because sampling has been geographically concentrated. There are regions where the contact signal could exist in ancient genomes and would not currently be detectable, simply because no one has sampled there yet. Future ancient DNA work along the coasts of Peru, Ecuador, and Colombia, and from island populations across the central Pacific, could either confirm the current model in finer resolution, or introduce complications that require substantial revision. The same is true for the Indian Ocean. The genetic history of Madagascar is well established at a broad level, but the precise timing of different migration waves, the degree of interaction between incoming Austronesian populations and East African groups, and the question of whether there were pre-Austronesian contacts between Africa and island Southeast Asia through Indian Ocean routes, these remain areas where the data is insufficient to support firm conclusions. Ancient DNA from sites around the Indian Ocean rim, including the Maldives, Sri Lanka, and the East African coast, could substantially change the picture. There is also an important ethical dimension to this research that shapes what can be known and how quickly. Ancient DNA analysis is a destructive process. Extracting genetic material from skeletal remains requires physically altering those remains, typically by drilling into a tooth or removing bone material. For many indigenous communities, this is not a neutral technical procedure. Human remains hold deep cultural and spiritual significance, and many communities have fought hard and long to repatriate ancestors taken by colonial-era collectors and museums. Asking those same communities to then permit further destructive analysis of those ancestors for academic research requires a level of trust and reciprocal benefit that has not always been offered and has not always been earned. Responsible researchers in this field have increasingly moved toward consent-based models where community involvement is built into study design, where findings are shared with descendant communities before or alongside publication, and where communities retain meaningful control over how their ancestors' remains are used. But this framework is uneven across the field, and the historical record of genetics research involving indigenous peoples includes enough examples of extraction without consent, misrepresentation of findings, and failure to return results, that skepticism from communities is not only understandable but appropriate. Some of the most genomically informative ancient remains in collections will not be sampled or should not be sampled until those relationships are built properly. That is a constraint on the pace of discovery, and it is the right constraint. What this means in practice is that the genomic map of ancient human contact will be filled in unevenly, with some regions and time periods becoming much clearer while others remain shadowed. It also means that findings in this field can run ahead of the ethical frameworks needed to contextualize them, which creates real risks. Genetic ancestry claims have been weaponized in harmful ways before, and a finding that a particular population has unexpected ancestry from a distant contact could be used by outside parties in ways that the community itself would not endorse. For researchers and audiences alike, perhaps the most important habit to cultivate is calibrated uncertainty. The evidence for Polynesian South American contact is strong. The evidence for the precise nature of that contact is weak. The evidence for pre-Columbian Norse presence in North America is strong. The evidence for extensive Norse-American interaction beyond a limited settlement is weak. Knowing the difference between these levels of confidence is not pedantry. It is the only honest way to engage with the research that is actively in motion. New data could overturn current conclusions in either direction. A well-dated ancient genome from coastal Chile could sharpen or complicate the Polynesian contact story. New analysis of Pacific Island populations might reveal multiple contact events where one is currently inferred. A discovery of pre-Columbian plant or animal remains with clear Atlantic African signatures could open a line of inquiry that currently lacks strong physical evidence. Conversely, a more rigorous dating of the chicken bones could close that particular question definitively. The frontier of this research is exactly that, a frontier. The strong findings are real, and they matter, but a significant portion of the most exciting questions in Pacific and Atlantic contact research remain genuinely unanswered, and the honest position is to hold those open questions as open, rather than rushing toward conclusions that feel satisfying before the evidence actually warrants them. 50 years ago, the dominant story of human exploration was relatively tidy. Continents were settled in a fixed sequence. Oceans were barriers. Contact between distant civilizations, when it happened at all, was accounted for by the recognized voyages of documented history. That story was always incomplete, and researchers who worked in archaeology and linguistics had been chipping away at its edges for decades. But it took genetics, and specifically the ability to read ancient DNA at scale, to make the full extent of that incompleteness visible in a way that was impossible to dismiss. What DNA has done is not replace the older tools of history. Archaeology still gives us the physical trace of human presence. Linguistics still maps the spread of language families across ocean distances. Botany still tracks the movement of domesticated plants. What genetics added was a way to see through time at the biological level, to ask directly whether populations that are now separated were ever in contact, and in some cases to answer that question with a precision that no artifact or text could offer. The result is a picture of ancient movement that is more connected, more daring, and more complicated than the version most of us were taught. The strongest examples from that picture deserve to be restated plainly. Polynesian navigators reached South America before Europeans ever crossed the Atlantic. That is not a hypothesis anymore. The genomic evidence across multiple Pacific Island populations carries a clear South American signal that predates European contact, and the distribution of the sweet potato across the Pacific provides independent botanical confirmation from a completely different evidentiary direction. These two lines of evidence, one genetic and one botanical, converging on the same conclusion without relying on each other, is exactly the kind of cross-disciplinary reinforcement that moves a finding from plausible to established. The Norse reached North America around 1,000 years ago, made landfall in Newfoundland, and established a settlement that archaeology has physically confirmed. That is not a saga embellishment. It is a documented historical fact. The Austronesian expansion across the Indian Ocean placed seafarers from Southeast Asia on the island of Madagascar. Thousands of miles from their origin point, in a migration that linguistics, genetics, and material culture all confirm. These are not peripheral footnotes to human history. They are among the most remarkable feats of navigation ever accomplished, and they happened long before they were officially recognized by the European scholarly tradition that shaped how exploration was defined and recorded. But precision matters here, and this is where the measured conclusion that the evidence actually supports has to be distinguished from the more dramatic version that is easier to tell. The genetic record shows contact, it does not always show sustained exchange. It shows movement, it does not always tell us who moved, in which direction, or how many times. The Polynesian South American contact is real, but the specific island of origin, the precise landing point on the South American coast, and the degree of back-and-forth interaction remain genuinely uncertain. The Norse were in North America, but whether they had extensive ongoing contact with indigenous populations beyond what the archaeological record at Lance Meadows implies is not established. A signal in a genome is evidence of contact. It is not a full account of what that contact looked like, what it meant, or how it shaped the populations involved. This distinction between evidence, inference, and myth is one that keeps coming up throughout this research and keeps mattering. The old myths were myths of isolation, stories that positioned certain civilizations as the first to venture into unknown territory, that drew sharp lines between the connected world and the uncontacted periphery. Those myths served particular interests at particular historical moments, and they persisted long after the evidence had started to complicate them. What the new genetics research offers is not a replacement mythology of an ancient, globally connected world where every civilization was in contact with every other. The evidence does not support that either. What it supports is something more interesting and more specific, that certain groups of people in certain periods crossed distances that conventional assumptions deemed impossible, left traces of those crossings in the genomes of their descendants, and then disappeared from the written record because the written record was not kept by them. The oceans were not empty corridors waiting for European ships to activate them. They were travelled. The Pacific in particular was navigated by people whose spatial knowledge, astronomical understanding, and boat-building capability allowed them to move across the largest body of water on Earth, with enough reliability to establish populations across thousands of islands. That this happened is no longer seriously in dispute. The genetic map of the Pacific confirms it in granular detail. What is still being worked out is the full extent of where those voyages reached, whether contacts were single events or recurrent ones, and what those contacts meant for the communities on both ends. There is more to learn. The ancient DNA record for large regions of South America, the Indian Ocean rim, and the Central Pacific is still incomplete. Future sampling will clarify some current uncertainties and will almost certainly introduce new ones. The honest position coming out of everything this research has produced so far is not that the map is now finished. It is that the map is now far more honest than it was. What has changed is the baseline assumption. The default is no longer isolation. The default for populations living near coastlines and capable of building ocean-going vessels is that contact was possible, that it was attempted, and in many documented cases that it succeeded. The burden of proof has shifted. The oceans were not walls. For the people who knew them best, they were roads, and those roads were used earlier, more widely, and more skillfully than the official story of human exploration ever gave them credit for.